Welcome back to the Deep Dive. If you feel like the life sciences industry is speeding up, like warp speed, you are absolutely right.
It really is.
The rate at which, lab breakthroughs are turning into actual approved treatments and commercial products has just accelerated past anything we've seen before.
It has.
We are standing right in the middle of a precision biotech revolution. And 2025 is really shaping up to be a pivotal year where all this theoretical potential becomes, well, real, tangible solutions.
It truly is an unprecedented moment. It's fueled by all these converging technologies.
Right.
We've sort of reached a critical mass where AI, automation, and these new molecular tools are all feeding off each other, creating this exponential growth.
You can measure that confidence, right?
You can. That confidence is measurable. There was a recent survey among global life sciences execs, and it showed 75% feeling explicitly optimistic about 2025.
Wow, 75%.
Yeah, and it's driven by strong growth expectations and, innovations that are solving actual real-world problems.
And that's our mission today. We've taken a deep dive into your stack of sources outlining the most impactful trends right now. Our goal is to synthesize these major forces, everything from gene editing and AI to how we grow food, and critically, how we govern our planet.
And we've organized around 7 major trends that are fueling all that optimism you mentioned.
Genetic engineering, AI, biomaterials, microbiome research.
RNA, high-throughput tech, and sustainability in biotech.
These aren't just isolated things anymore, are they?
Not at all. They are the integrated components of a single, powerful technological wave. It's fundamentally reshaping how we live, how we heal, and how we interact with the environment.
So we are starting where the action is most visible and, frankly, most profound. Gene editing.
Makes sense.
This is where we see that immediate, measurable transition from basic research into mainstream health and agriculture. This revolution starts at the smallest possible scale, but its impact is just enormous.
It is.
Okay, let's unpack this, starting at the molecular level.
Let's do it.
When we talk about CRISPR technology, which just a few short years ago was still, stuck in research papers, we have to recognize the massive, immediate, historical milestones.
Yeah.
The biggest one has to be the December 2023 FDA approval of Casgevy.
Oh, absolutely.
This wasn't just an approval. It was the world's first FDA-approved therapy using the CRISPR-Cas9 genome editing technology.
Right. And that set a precedent that fundamentally changes the entire regulatory landscape for genetic medicine.
The significance here, you can't overstate it, can you?
You can't, because Casgevy is approved for a chronic, debilitating inherited disorder, sickle cell disease, or SCD.
Right.
It targets patients 12 and older, specifically those dealing with recurrent vaso-occlusive crises. Those are the painful blockages that really define the severity of the disease.
So this is precision medicine delivering a cure, not just managing symptoms.
That's the key, a curative solution.
How does Casgevy actually use CRISPR to fix a blood disorder that affects millions of people around the world? Walk us through the mechanism, because this is where the precision part is just incredible.
Okay, so the process is elegant, but pretty complex. It involves ex vivo engineering, so that's all done outside the body.
Got it.
It modifies the patient's own hematopoietic stem cells, which are basically their blood-making factories in the bone marrow.
Okay.
Scientists harvest these cells, and in the lab, they use the CRISPR-Cas9 system. And you have to remember, the Cas9 enzyme acts like molecular scissors.
The famous cut in the DNA.
Exactly. It's directed by a guide RNA to a very specific DNA sequence. In the context of Casgevy, they are cutting the DNA to disrupt a gene called BCL11A.
And what does that gene do normally?
It normally acts as a brake. It represses the production of fetal hemoglobin, or HbF. So once that brake is removed by the precise CRISPR edit, the modified cells, when they're transplanted back into the patient, they graft in the bone marrow and start ramping up the production of HbF.
Fetal hemoglobin, which is normally suppressed shortly after we're born, acts as a protective mechanism. It's a natural thing we already have.
Correct. And the elevated sustained levels of HbF are structurally different from adult hemoglobin. They effectively prevent the sickling of red blood cells.
Which is the root cause of all the pain and organ damage.
Root cause. And because these are the patient's own stem cells, this is designed as a one-time treatment delivering a durable, potentially curative effect.
That shift from decades of just palliative treatment to a one-and-done curative gene edit is transformational.
It is.
And this momentum isn't just in the U.S., is it?
Not at all. The European Medicines Agency, the EMA, has mirrored this progress. In early 2024, they approved a related gene editing therapy, exagemglogin autotemcel.
Okay.
And significantly, that therapy targets both sickle cell disease and transfusion-dependent beta thalassemia, which is another inherited blood disorder.
So the dual approval.
Okay, a dual approval. It demonstrates a global consensus on the safety and efficacy of this specific type of genetic engineering, moving the tech swiftly from a lab curiosity to a global clinical standard of care.
The power of CRISPR isn't limited to correcting inherited blood disorders.
Yeah.
It's also being rapidly applied against complex acquired diseases like cancer.
Right.
And that's where we see precision editing really shine like an arrow that hits only the bullseye by targeting the cancer cell's specific defense mechanisms.
That's the critical shift. The focus is moving beyond just killing cells and more toward how gene editing can be used to resensitize cancer cells,
Making old treatments work again.
making traditional treatments that have stopped working effective again. This opens up entirely new treatment pathways for advanced cancers.
I'm looking at a fascinating finding from November 2025 from ChristianaCare's Gene Editing Institute about lung cancer. They found that disabling a specific gene, NRF2, with CRISPR could actually reverse a tumor's resistance to chemotherapy.
Which is a game changer.
It's a huge game changer because resistance is so often why treatments ultimately fail.
NRF2 is arguably one of the most important genes in understanding tumor resilience. It stands for nuclear factor erythroid 2 related factor 2.
Okay.
You can think of NRF2 as the cell's internal general that controls the entire suite of stress response mechanisms.
So when the cell's under attack from chemo or radiation,
Exactly. When it's under attack, NRF2 activates hundreds of defensive genes that neutralize the threat.
it's the master switch for cellular defense.
Precisely. In many aggressive solid tumors, particularly lung squamous cell carcinoma,
Which is pretty common, right?
it accounts for up to 30% of all lung cancer cases. In those tumors, a specific mutation, R34G, causes NRF2 to become permanently overactive.
So the general's always mobilized?
It's like the general has been permanently mobilized even when there's no threat. It creates a fortified cell before treatment even begins.
And when it's permanently fortified, it helps the cancer cells withstand the chemical attack from drugs like carboplatin and paclitaxel.
That's exactly right. The ChristianaCare researchers took lung cancer cells with this R34G mutation and used CRISPR-Cas9 to essentially knock out or silence the NRF2 gene.
They just switched it off.
By disabling this overactive master regulator, they successfully restored the cancer cell's sensitivity to the chemotherapy drugs.
And in animal models.
In animal models, the tumors that were directly treated with this CRISPR approach grew much more slowly and responded dramatically better to the standard chemical regimen.
The implication here is just enormous. If you can resensitize a tumor, you extend the utility of decades-old established drugs.
That's a major win.
It's a huge win for patients whose cancers have become refractory.
It is. And the underlying principle targeting overactive NRF2 is not exclusive to lung cancer. This same mechanism of resistance applies across a wide range of treatment-resistant solid tumors.
Like what?
Liver, esophageal, and head and neck cancers, for example. This opens up the possibility of developing a gene-editing adjuvant, a secondary treatment.
That you use alongside chemo.
Exactly. To improve outcomes and potentially allow patients to tolerate and benefit from treatment for much longer. The focus is shifting from what new drug can we invent to how can we reactivate the drugs we already have.
What's so fascinating here, even as we're celebrating this massive clinical success of standard CRISPR, the toolkit itself is already evolving.
Right.
Scientists are moving beyond that first generation to what we can think of as the second and third generations of precision editing, base editing, and prime editing.
That's a crucial point. Standard CRISPR-Cas9 works by creating a double-strand break in the DNA helix.
A full cut.
A full cut. And while it's directed, the cell's natural repair mechanism that tries to fix that break often introduces small random insertions or deletions. We call them indels.
And those are not always what you want.
Right. They're useful for knocking a gene out, but they lack true surgical precision. Base and prime editing aim to eliminate the need for that double-strand break entirely. They offer atomic-level control.
So let's start with base editing. If standard CRISPR is a pair of scissors, what's the analogy for base editing?
Base editing is like a chemical eraser and a pen.
Okay.
It allows you to change a single letter in the genetic code, a substitution, without severing the DNA backbone.
How does it do that?
Base editors consist of a Cas9 enzyme that's been modified so it's inactive, or it only cuts one strand, we call that a nickase, and that's fused with a deaminase enzyme.
And the deaminase enzyme, that's the pen that does the chemistry.
Exactly. This fusion allows for direct conversion of one nucleotide base pair to another in situ, right there on the strand, without that messy double-strand break.
So no need for the cell's messy repair process.
Right. And we differentiate between cytosine base editors, or CBEs, which convert CG pairs to TA, and adenine base editors, ABEs, which convert AT to GC.
And that lets you correct a huge number of diseases caused by just a single typo in the DNA.
A huge number of disease-causing port mutations, yes.
I see mentions of second-generation editors being enhanced to make them even better. What was the limitation they were trying to fix?
The initial limitation was efficiency and a bit of collateral damage. In the earliest CBEs, the cell's own repair mechanism sometimes tried to reverse the edit.
To undo the work.
Right, or it introduced small, unwanted deletions near the target site. So researchers address this by adding components like uracil DNA glycosylase inhibitor, or UGI, to the second generation.
And UGI does what?
It inhibits the base excision repair pathway. By blocking the cell's attempt to undo the work, they significantly improve the efficiency of the C to T conversion and drastically reduce the risk of indel formation. It enhances precision to, close to 100%.
If base editing is targeted substitution, prime editing sounds even more versatile, like the ultimate search and replace function in a word processor.
That's the perfect analogy. Prime editing is considered the next frontier for ultimate precision. It combines the Cas9 nickase with a reverse transcriptase enzyme.
Okay.
And critically, it uses a unique guide RNA called a PEG RNA, which not only directs the editor to the target, but also carries the desired sequence change.
So instead of just finding the cut point, the guide RNA actually brings the new blueprint with it.
Precisely. This allows prime editing to enable precise insertions, deletions, and all 12 types of substitutions, even large chunks of sequence.
So it's way more versatile than CRISPR or base editing.
Significantly more versatile for complex genetic repairs where you need to move a small paragraph of code, not just change a letter.
Is this technology stable enough for immediate use, or is it still mostly in research?
It's seeing very rapid research deployment. The challenges now are making it efficient across all cell types. In plants, for instance, researchers are working to improve PE systems using variations like PE6C.
Okay.
And they're also using techniques like conditional knockdown of the OSMLH1 gene in rice.
And why would they do that?
Because improving the natural repair machinery can make the prime editor more efficient in incorporating the new sequence without, disturbing the plant's fertility. The research is very focused on translating this versatility into human therapeutics.
If these tools are so precise, what is the major technical hurdle they still face, despite all these generational advances?
The single biggest persistent challenge remains off-target effects.
Unintended edits.
Unintended edits occurring at sites other than the intended target. While base and prime editing reduce the risk associated with double-strand breaks, they don't eliminate the risk of the editor binding or editing at a similar but wrong sequence elsewhere in the genome.
And for human therapies, that's a huge problem.
For human therapeutics, even a tiny percentage of off-target edits could lead to serious adverse effects, even cancer.
So how are researchers addressing this critical specificity challenge? What are the mitigation strategies?
They're using multiple sophisticated strategies, and they rely heavily on engineering. First, they're developing high-fidelity CRISPR-Cas9 nucleases. These are engineered Cas9 proteins that are just much pickier about where they cut. Second, they're engineering Cas9 variants with altered PAM specificities. The PAM is the short DNA sequence Cas9 has to recognize before it cuts. By changing the required PAM sequence, they can dramatically increase specificity and limit the accessible targets.
And this brings us back to one of our core trends, AI.
Yes, they are crucially integrating AI and machine learning to predict which guide RNAs will be most efficient and least likely to cause off-target activity.
So the AI acts as a pre-screening tool.
An intelligent pre-screening tool for every potential genetic experiment. Deep learning methods are being rigorously benchmarked specifically to predict on- and off-target CRISPR-Cas9 activity across the entire genome.
The pace of innovation we've just discussed relies so heavily on speed, and nothing demonstrated speed in the last few years quite like mRNA vaccines.
Not even close.
That success radically transformed the perception of RNA technology, and this innovation stream isn't slowing down. It's moving way beyond infectious disease and into oncology and chronic disease.
That's a major force driving the optimism for 2025. The realization is that if RNA can instruct our cells to produce a harmless viral protein to train our immune system,
It can be programmed to produce almost any protein.
any protein, an antibody, an enzyme, an immunomodulator, this adaptability is leading researchers to develop therapies for complex diseases like cancer, genetic disorders, and autoimmune diseases. It's taking RNA research into completely new therapeutic avenues.
What specific innovations are standing out in the RNA field that have the potential to solve some of the logistical bottlenecks we have in global health?
One of the most promising is self-amplifying RNA, or SARNA.
Yeah.
This technology is a significant step forward because the RNA molecule, once it's inside the cell, carries instructions to replicate itself.
So you need a much smaller dose.
Exactly. You require significantly smaller doses to achieve the same therapeutic effect compared to non-amplifying mRNA.
That immediately addresses global distribution, doesn't it?
Without a doubt. Smaller dose requirements dramatically reduce manufacturing costs and reliance on vast amounts of raw materials. And more importantly, it's invaluable for logistics, especially for mass vaccination campaigns or treatment distribution in resource-limited settings.
Where maintaining a huge, complex cold chain is basically impossible.
Impossible. SARNA promises greater efficiency with much less physical burden.
And then there are those fascinating stable molecules, circular RNAs. They sound like the anti-mRNA designed for permanence instead of rapid degradation.
They are structurally unique. Unlike linear RNA molecules, which are easily degraded by enzymes in the cell, circular RNAs, or circRNAs, have a closed-loop structure.
Which makes them super stable.
Highly stable and long-lasting. They were once dismissed as junk RNA, but we now know they were crucial regulators of gene expression.
And how is that stability being used therapeutically?
Their stability makes them excellent emerging therapeutic targets and biomarkers. Because they are often dysregulated in disease states, circRNAs are being specifically linked to complex conditions like cancer and various cardiovascular conditions.
So they can be used for diagnostics.
For earlier, non-invasive disease detection, and potentially using them as highly stable therapeutic vectors themselves for sustained expression of a drug target.
This expansion of therapeutic modality is driving huge shifts in the pharmaceutical market, particularly in the cell therapy space, like CAR-RT treatments for cancer.
Right.
And we see a fascinating contradiction here. Some major players are retreating, but at the same time, others are making massive strategic acquisitions. What's driving this split?
The market dynamics are complex. They reflect the high risk and high reward of cell therapy. We saw a general pullback from some major pharma companies, like Takeda and Novo Nordisk, abandoning some long-term, high-cost investments in the field.
Probably due to scaling challenges and ROI hurdles?
Exactly. But this hesitation is immediately juxtaposed against significant, high-value M&A deals by others who see the long-term transformative strategic value.
So it's a segmentation.
It's just a segmentation. Some are retreating from ex vivo complexity, while others are buying into next-generation simplicity.
Let's drill down on that next-gen simplicity. Bristol-Myers Squibb, or BMS, made headlines in 2025 by dropping $1.5 billion to acquire Orbital Therapeutics. Why was Orbital such a prize?
BMS acquired Orbital specifically for their circular RNA therapy pipeline led by OTX-201. This deal wasn't just about adding a single drug, it was about acquiring a capability.
The ability to manufacture therapeutic constructs in vivo.
In vivo, OTX-201 is designed to induce a patient's own cells via circular RNA instructions to manufacture anti-CD19 CAR constructs inside the body.
Wait, in vivo means inside the body. That is a fundamental paradigm shift for CAR-T therapy.
It is.
Traditionally, CAR-T has been ex vivo, right? You have to harvest cells, engineer them in a huge, expensive, centralized lab, quality check them, and then reinfuse them days or weeks later.
And that process is slow, geographically limited, and incredibly costly, often $500,000 per patient.
That complexity and cost are exactly what the Orbital deal targets.
Exactly. The in vivo approach bypasses the entire specialized logistics and manufacturing infrastructure currently required. The orbital therapy is initially focused on treating B-cell-driven autoimmune disorders, but the platform capability is the real prize.
And this shift toward in vivo engineering is gaining traction in oncology, too, where the need to reduce costs and turnaround time is even more urgent.
Absolutely. Early in 2025, the first patient was dosed in the Biotech trial for ESO-T01.
What's that?
It's the first in vivo BCMA-directed CAR-RT therapy specifically targeting relapsed or refractory multiple myeloma. The goal is revolutionary: to reprogram the patient's T cells to attack cancer inside their body using a simple, potentially off-the-shelf injection.
If the logistical benefits are so massive, why did it take until 2025 for the first patient to be dosed in an in vivo CAR-2 trial? What technical barrier just fell?
The primary technical barrier was delivery and efficiency. Getting a gene editing tool or an RNA instruction package safely and efficiently to the right cell type. In this case, T cells inside a complex human body is immensely difficult.
Right.
Traditional delivery methods like viral vectors have safety limitations. This recent progress is tied to vastly improved lipid nanoparticle LNP delivery systems.
The same tech from the mRNA vaccines.
The same technology perfected during the pandemic. LNPs can now be engineered with higher precision and lower toxicity, making the in vivo delivery of the genetic instructions finally viable and scalable.
So this is a beautiful example of cross-pollination. The LNP improvements driven by vaccine research are now fueling a cost revolution in advanced cancer therapy.
Precisely. Biotech suggests that this in vivo approach could potentially lower the cost of treatment by an entire order of magnitude compared to current ex vivo CRT treatments.
A tenth of the cost.
And if successful, that makes these life-saving therapies accessible to many more people globally and fundamentally changes the profitability models for cell therapy companies.
Alongside these groundbreaking in vivo shifts, we've also seen traditional replacement gene therapy treatments gain crucial regulatory approval for rare diseases.
That's correct. In November 2025, the EMA recommended Waskyra, which is the first gene therapy approved to treat the rare inherited Wiskott-Aldrich syndrome.
A debilitating disease.
Very. It primarily affects males, characterized by immunodeficiency and bleeding issues. These specialized approvals for conditions that previously had limited options just show the sustained progress.
And in terms of big picture strategy, the pharma giants are clearly making large bets on the intersection of gene editing and high-volume chronic diseases. Eli Lilly, for example, invested up to $1.3 billion in acquiring Verve Therapeutics.
That acquisition was highly strategic, focused on in vivo gene editing programs specifically for cardiovascular disease.
And what's their lead target?
Their lead asset targets the PCSK9 gene. This gene is closely linked to LDL cholesterol levels, the bad cholesterol. Normally, PCSK9 degrades the receptors that clear LDL from the bloodstream. Verve's approach is to use gene editing to permanently silence the PCSK9 gene in liver cells, leading to lifelong reductions in LDL and, theoretically, lifelong protection against heart disease.
So instead of a daily pill or an injection every few weeks, it's a one-time genetic intervention for a massive public health crisis.
It is the ultimate preventative medicine, delivered via precision editing. These acquisitions aren't about minor drug pipeline additions. They are about acquiring fundamental, transformative capabilities that reshape the business model of medicine entirely.
Moving toward true personalized medicine at scale.
That's the goal.
Which brings us back to one of the overarching trends you mentioned earlier.
Yeah.
The role of AI. We're seeing a powerful convergence where AI isn't just analyzing clinical trial data, it's the engine accelerating the precision biotech revolution itself.
AI is the essential force multiplier here, particularly when you're dealing with the complexity of biological systems and the need for precision. It is speeding up every stage: drug discovery, refining diagnostics, and profoundly revolutionizing protein design. Well, consider the challenge of finding a small molecule or a protein fragment that precisely interacts with the disease target. A human researcher or even high-throughput screening might test thousands.
But AI can model millions.
Exactly. A great example is the system developed at MIT, which uses deep learning models to predict stable, functional protein fragments. This AI can predict which fragments can bind to or inhibit a specific target protein.
And that reduces years of lab work down to days.
It significantly accelerates the identification and optimization of therapeutic compounds, reducing what used to be years of wet lab iteration down to days of computational modeling.
So AI helps decide where to cut and what protein fragment to use, while base and prime editing give us better tools for how to perform that precise change.
And improved LNPs allow us to deliver the instruction. It's an interconnected system.
Precisely. And this synergy is why the optimism among life sciences executives is so high. AI integration is leading to faster, more targeted, and crucially, more personalized solutions across the entire therapeutic pipeline.
And that's driving innovation in areas like CAR-T delivery and cardiovascular gene editing.
If biotech is transforming human health at warp speed, it is equally essential in transforming our ecosystems, particularly agriculture. We face immense global challenges in securing our food supply, and Time is running out.
The stakes couldn't be higher. Forecasts suggest we need to nearly double global crop production by 2050 just to feed a growing population.
But we have to do that while dealing with climate change.
Simultaneously, we are confronting existential threats from climate change, rising temperatures, erratic rainfall, and severe abiotic stresses like persistent drought, excessive heat, and high soil salinity.
And traditional breeding is just too slow.
It's far too slow. Relying on traditional breeding methods, which take years or even decades, simply will not be fast enough to adapt our crops to these rapidly changing environments.
So how is precision gene editing CRISPR, base editors, prime editors, responding to this climate crisis in the fields?
Precision editing offers an unprecedented shortcut. It is used to quickly and efficiently manipulate key regulator genes within crops. These are the genes responsible for coordinating the plant's entire stress response system.
The ones that traditional breeding struggles to isolate without bringing along unwanted traits.
Exactly.
Can we break down a few of those regulator genes and explain what they actually do for the plant?
Certainly, scientists are targeting genes like D or E, the dehydration responsive element binding protein. D or E acts like a master faucet, turning on the plant's entire water conservation and drought response system.
So you make the plant more drought tolerant.
Much more. Then there are HSP genes, heat shock proteins, which act as molecular chaperones, preventing cellular damage when temperatures soar.
And the salt tolerance challenge.
That's handled by genes like SOS, which stands for salt overly sensitive. They manage ion homeostasis, basically regulating how the plant controls its internal salt levels.
Which is critical in areas with bad irrigation or rising sea levels.
Right. And finally, genes like Erecta impact the shape and structure of the plant, improving canopy architecture and dramatically boosting water use efficiency.
So instead of waiting 10 generations for a natural mutation, we can make that precise climate-resilient edit in a single cycle.
Exactly. And this isn't future research, it's commercial reality. We saw the world's first CRISPR-edited food enter the Japanese market back in 2022.
The GABA-enriched tomato.
That product serves as proof of concept for the speed and efficacy of the technology in a commercial food setting. That was achieved within just a few years of the initial research.
Let's talk about the business side of this. That early success suggests massive market growth is coming for gene-edited food. What are the projections?
The data supports robust investor confidence. The gene-edited food market is poised for exponential growth, driven not just by climate necessity, but by the demand for sustainable, high-yield, and nutritious food.
And the overall value?
The overall market value is estimated to reach over $3.5 billion by 2033. This specific dollar figure tells us that investors see this technology as de-risked and scalable.
They see it less as experimental science and more as a necessary agricultural input?
Exactly.
And within that $3.5 billion market, what are the dominant growth segments?
There are two key segments driving the growth. First, the plant segment is anticipated to dominate. It's projected to account for over 70% of the market value by 2033.
Which makes sense. Staple crops are the backbone of the food supply.
Right. Rice, wheat, corn, and soy. Innovations here prioritize massive-scale yield increases and broad pest and disease resistance.
And the second segment.
Second, the commercial application segment will drive the bulk of the market growth, estimated at over $2.8 billion by 2033. This is crucial for understanding adoption.
Why the commercial dominance? Is this being pushed by consumers or by the producers?
It's overwhelmingly driven by large-scale agricultural producers and food processors. They're the early large-volume adopters because they are better positioned financially to invest in these higher-tech seeds.
And the payoff is immediate.
Immediate and significant. Increased efficiency, higher profit margins, and drastically reduced input costs for things like water, fertilizer, and pesticides. This rapid commercial adoption is what makes sustainable agriculture scalable.
It's driven by economic incentives as much as environmental needs.
Absolutely.
Beyond staple crops for bulk efficiency, precision editing allows for more niche, specialized trait engineering products that directly benefit the consumer or tackle specific public health issues. What are some of those?
The diversity is remarkable. We see examples like vitamin-enriched rice designed to combat dietary deficiencies in developing nations.
Sure.
There are fruits engineered for improved shelf life, which addresses the estimated 30% of global food waste that happens post-harvest.
And what about allergens?
There's specialized work focused on allergen mitigation, like hypoallergenic milk or peanuts, which could alleviate severe public health burdens. The market for these specialized gene-edited food products alone is projected to reach over $2.1 billion by 2033.
And just like in human health, AI is acting as a force multiplier here, driving precision farming technology and the move toward more sustainable inputs.
Absolutely. The convergence is complete. AI is fundamental to creating adaptive inputs and streamlining processes. Companies like INRI are using AI to guide precision gene editing to fine-tune traits like nitrogen efficiency and drought tolerance.
What are their targets?
They are explicitly targeting up to 20% yield increases coupled with 40% reductions in nitrogen and water use. That's a huge sustainability win that also saves farmers money.
And we are seeing AI applied directly in the field, literally at the plant level, changing how chemicals are used.
Yes. John Deere's See and Spray AI technology is a prime example, instead of broadcasting herbicides across the entire field.
Which is a huge source of runoff and resistance.
Right. The AI uses computer vision to identify weeds in real time and selectively spray only those specific plants. This is reducing herbicide use by up to 90%.
90%, that's incredible.
It cuts costs and greatly reduces the environmental footprint. Other firms are deploying predictive tools and integrated farm management systems, making AI a co-pilot for farmers.
This overwhelming shift towards sustainability is also fueling explosive growth in the biological inputs market, moving away from synthesized chemistry toward engineered biology.
That market is seeing massive investment. The market for biological inputs, biofertilizers, biopesticides, biostimulants, is projected to grow at a 12% compound annual growth rate, reaching a staggering $115 billion by the 2040s.
$115 billion. That is a truly massive figure. What is driving that scale of growth?
Three main drivers. One, deep environmental concerns and stricter regulatory scrutiny of chemical inputs, particularly in Europe. Two, the rising problem of pesticide resistance.
Forcing farmers to seek new solutions.
Exactly. And three, the realization that beneficial soil microbes, the microbiome, are essential for crop health.
And how are they finding these new biologicals?
Companies are now leveraging AI and high-throughput screening to screen massive microbial libraries for new biopesticides and biostimulants.
So AI can predict which microbes will work best in which soil.
It can predict which microbes will survive, thrive, and deliver specific benefits in specific soil environments, making the application of biological inputs more predictable and effective than ever before. This synergy is the engine driving this market toward $115 billion.
Now we shift from editing existing life to building new biological capabilities entirely. This is where synthetic biology, or synbio, takes center stage. This field, which blends biology and engineering, is rightly being called the factory of the future.
That's the most accurate description. Synbio is essentially programming living cells, microbes, yeast, or algae to act as customizable production factories.
So instead of building a petrochemical plant to produce a chemical,
you program a yeast strain to produce that same compound or a novel material, often using sustainable, renewable inputs like sugar instead of oil.
And the economic projections suggest this is moving out of the academic lab and into industrial infrastructure.
Absolutely. The field is projected to grow tenfold, potentially reaching $100 billion by 2030. Synbio is already creating sustainable outputs like pharmaceuticals, renewable biofuels, and lab-grown meat.
You mentioned high-throughput technologies earlier, and I think this is where that missing piece of the blueprint really integrates. How does automation and screening speed up the Synbio factory design process?
It's fundamental to scaling Synbio. Designing a microbial factory is iterative. You have to try millions of combinations of genetic pathways to find the one that works efficiently.
And you can't do that by hand.
No way. High-throughput screening involves robotically testing thousands or even tens of thousands of different engineered cell lines simultaneously under various conditions.
And AI interprets the data.
AI interprets this massive data flow, predicting which genetic modifications or designs are most likely to succeed. This convergence reduces the typical months of trial and error to weeks.
Which is essential for going from the lab bench to industrial manufacturing.
Absolutely.
What are the significant hurdles that Synbio still faces, even with this immense industrial potential?
The hurdles are real, and they are why scaling to that $100 billion market's challenging. You have significant ethical challenges regarding the creation of new life forms, ecological risks, security concerns.
But what about on the commercial side?
On the commercial side, the biggest hurdle remains scaling barriers. Getting a successful process from a small bioreactor in a lab to a 100,000-liter industrial tank often introduces new variables, contaminants, and efficiency drops that are difficult to predict.
The push for sustainability isn't just about food. It's about broader environmental solutions. And Synbio is tackling some of the toughest industrial challenges, like pollution control and material science.
Biotech is now driving the creation of genuinely eco-friendly solutions to replace petroleum-based materials. This is where the last missing trend, biomaterials, becomes critically important.
So what are we talking about here?
We are seeing bio-based solutions moving toward commercial viability, such as new biodegradable plastics produced by engineered bacteria or yeast. These aren't just slightly greener versions of existing materials. They are novel polymers designed from the molecule up to degrade harmlessly.
Or possess superior strength, like synthetic spider silk.
Exactly, produced in a bioreactor.
So we're using biology to solve the problem of biological waste.
Precisely. And that focus on environmental impact also includes research into direct climate intervention and carbon management, specifically in carbon capture and utilization.
Okay.
Researchers are advancing work on engineered organisms, often highly efficient algae or bacteria, that are designed to capture atmospheric carbon dioxide, or TXTO2, far more efficiently than natural systems.
And then convert it into something useful.
They then convert that captured TXTO2 into valuable, economically viable products.
That's a powerful two-pronged approach.
It is. The conversion can result in useful materials, like new renewable biofuels, bioplastics, or even food ingredients like single-cell proteins. It offers a dual benefit, combating climate change while creating new, sustainable economic materials.
We're also seeing biotech tackle existing environmental damage through bioremediation.
This leverages the power of microbiome research. Scientists are engineering microbial communities, often incorporating extremophiles.
Microbes that live in really harsh environments.
Right, like deep sea vents or highly acidic hot springs. They're using them to clean up industrial pollution, literally degrading plastics in landfills, remediating large oil spills, and detoxifying heavy metals.
Now, here's where it gets really interesting. If these biological and climate solutions cross so many different areas, agriculture, manufacturing, pollution, medicine, the governance of them must be incredibly complex.
It is.
Your sources point to a critical policy challenge raised at the United Nations Environment Assembly, or UNEADU 7, in December 2025.
This is the crux of the policy problem when technology moves this fast. The challenge identified at UNEA 7 was profound. Stubborn silos are hindering governmental action.
What does that mean exactly?
The key realization is that nature and the compounded crises affecting it, climate, biodiversity loss, pollution, do not recognize the artificial lines between government ministries.
So the left hand doesn't know what the right hand is doing.
Basically, if the Environment Department is tackling biodiversity loss, but the Treasury Department is funding unsustainable infrastructure, the solution fails.
So the technology is forcing us to acknowledge that our government structures are decades behind our scientific capabilities. We've been trying to solve integrated, complex problems using compartmentalized, siloed budgets and policies.
Precisely. We're dealing with the tapestry of multilateral environmental agreements, or MEA's global accords, like the Paris Agreement on Climate, the Convention on Biological Diversity.
And they're all handled by different departments.
Often implemented by different government ministries, leading to duplication, contradiction, or simply a lack of necessary funding.
What is the proposed solution to this silo problem? And what does it mean to integrate this into macro-fiscal planning?
The policy solution being advocated is whole of government and whole of society planning. It's a radical concept. It means environmental agreements must be integrated directly into macro-fiscal planning.
So national budgeting, treasury policy.
National budgeting, central bank risk assessment, infrastructure spending. It means the Minister of Finance must treat nature loss and climate risk as financial liabilities that have to be budgeted for, mitigated, and invested against, just like national debt or currency fluctuation.
It moves nature from an isolated environmental issue to a core economic stability issue.
That's the powerful shift. It's a deliberate effort to break the silos. It also requires integrated data systems to ensure consistency and accountability across policy areas.
Are there specific examples of countries actually starting to do this?
Yes, several countries were highlighted as leading the way. Panama, for instance, has its Nature Pledge, which consolidates all its policy processes into a single document.
Okay.
Additionally, countries like Rwanda, Costa Rica, Colombia, and Mongolia are working to align their national biodiversity strategies, their climate targets, and their land degradation targets using common baselines, indicators, and monitoring systems.
A unified landscape plan.
Exactly. It's how global obligations are being turned into impactful, country-driven solutions that also deliver economic benefits.
We've covered the power of gene editing in medicine and agriculture, but with this power comes immense responsibility and deeply complex ethical questions. The arrival of human gene editing therapies like Casgevy necessitates continuous engagement on ethical and societal discussions.
That is the essential constraint on this rapid progress. Continuous basic research must proceed alongside robust ethical review. Collaboration isn't just a suggestion. It is essential across human, life, biomedical, legal, and social sciences.
I found a fascinating detail in the sources about a Swiss project that's carefully evaluating the potential use of CRISPR in the human germline.
Right, editing genes that can be inherited.
And they are using a unique, ethically responsible animal model.
This specific project perfectly illustrates how scientists are trying to gather crucial data without crossing irreversible ethical lines. They are using superfluous oocytes, eggs from pigs and cattle that are normally just discarded by slaughterhouses.
So they're not creating new embryos just for research.
No. These discarded animal cells provide an accessible, ethically responsible model for germline research. They are using them to gain insights into the long-term risks, efficiency, and unintended consequences of germline editing before anyone attempts to move toward a human context.
That's a highly practical way to address a massive ethical minefield.
And this leads to the broader conversation about regulation. What is the scientific community's stance on regulating these powerful tools? Do they want to regulate themselves?
The sources indicate that scientists working on gene editing actually express a preference for interdisciplinary professional engagement to strengthen regulation.
So not self-regulation.
Not solely self-regulation. They are keenly aware that technical expertise alone is insufficient for governing tools with such profound societal implications. They see collaboration with legal and social science experts as necessary.
They recognize that public trust is fragile, and the speed of innovation must be matched by the robustness of its governance framework.
To bring in a crucial policy parallel that demonstrates the danger when technology moves too quickly and lacks a consensus governance structure, we should discuss the high-stakes debate around solar geoengineering.
The one that flared up at UNA7.
Right. It's not a biotech trend, but it perfectly illustrates the governance crisis for powerful, new global technologies.
It's a perfect comparison. It shows the difficulty of regulating global technological reach, especially when the consequences are irreversible.
So we're talking about modifying the atmosphere to reflect sunlight and cool the planet.
Exactly. And it was discussed intensely on the margins of the December 2025 UNA7 meeting, highlighting growing political momentum against deploying such a highly unpredictable and ungovernable technology.
And this dialogue resulted in a significant proposal from the Africa Group, pushing for a plurilateral dialogue on a solar geoengineering non-use agreement.
A preemptive policy move to halt a technology before it truly takes off.
That's the strategic goal, preemption.
The proposal is based on 5 strict pillars designed to prevent dangerous technology development and deployment globally. And they reflect deep-seated fears about equity and control.
What are those 5 core principles?
First, no public funding for the research. The fear is that public investment legitimizes and fast-tracks a technology that lacks consensus. Second, no outdoor experiments.
To prevent accidents or unilateral deployment.
Exactly. Third, no patents on the technology. This is crucial for equity. The fear is that a single corporation or nation could gain intellectual property control over a global climate lever.
That's terrifying. A single entity owning the solution to global warming.
Indeed. The 4th pillar is no deployment whatsoever, a hard stop. And the 5th is no support for the technology in international institutions. This ensures bodies like the World Bank don't inadvertently fund or promote its adoption.
This political movement underscores the fact that when a technology has irreversible global consequences, whether it's atmospheric modification or human germline editing, policymakers have to move fast.
The speed of the policy debate must try to match the speed of the technological development.
So what does this all mean? We have witnessed a powerful convergence in 2025. AI is now accelerating the precision of gene editing, driving tools like base and prime editing to near-surgical accuracy.
While also making cell therapy delivery cheaper and more accessible through in vivo innovation.
This precision is fueling massive commercial breakthroughs in oncology, rare diseases, and creating a multi-billion-dollar market for climate-resilient agriculture and sustainable biomaterials via synthetic biology.
And the integration of high-throughput technology ensures that the molecular design phase is rapidly scalable to industrial levels.
This technological momentum is forcing critical conversations about how we govern these advancements, whether through integrated whole-of-government approaches, breaking those stubborn silos, or through deep interdisciplinary engagement on ethics.
We are seeing a fierce race between technological capability and our collective ability to regulate it safely, equitably, and ethically. We have seen the incredible speed of precision gene editing move from the lab bench to a $3.5 billion commercial food market and an FDA-approved drug for sickle cell disease in record time.
Right.
Yet the ethical discussion around human germline editing, the ultimate act of biological precision capable of affecting generations, remains highly complex and deliberately slow.
And the research community itself is advocating for rigorous, externally driven regulation.
They are, recognizing the gravity of it all.
This leaves us with a provocative thought that underscores the core challenge of this decade. We have the tools today, right now, to genetically engineer our children. We have the capability to permanently alter global climate systems.
We do.
How quickly can our collective ethical and regulatory frameworks mature to govern these transformative tools before the technological momentum fueled by incredible speed and immense profit potential makes the decision for us?
That's the critical tension point to keep watching as we move forward.
Thank you for joining us for the deep dive.